Okay, thank you very much for inviting me to give this talk. Can you hear me okay? Yes?
Yes.
Yeah. I was asked to do something around the vaccines and testing, etc. So I'm going to really, it's more on the technical aspects of these that people were interested in. So I'm going to try and cover these, and I apologize if I'm going to start from quite basic principles because I think it's maybe quite a wide audience. So if anybody, if I offend anybody, please go and get a cup of tea or something. Right, can I have the first slide, please, Jonathan? Thank you. So you're all familiar probably with seeing pictures of the coronavirus by now. So I'm just going to look a little bit more at the structure because this will be important on the way going through. So this is an envelope virus, and you're all familiar with the spike protein by now on the outside of the virus. And this protein attaches the virus to the cell, and this particular, the SARS-2 virus, which is the one which causes COVID-19, uses the angiotensin converting enzyme 2 receptor, or ACE2. This is a feature of viruses that they hijack proteins or sometimes other molecules on the cell surface to allow them to get inside the cell. So these are normally molecules which have normal particular cell functions, but the virus actually takes them over as a means of transport into the cell. And the spike protein, we'll come back to it later, but it is the important one for protective antibody called neutralizing antibody, and it's the body making that kind of antibody. The other, I was hoping to point at this, but I can. The other ones are proteins of importance. I'm just going to point out, not through them all, is the nucleocapsid protein. And the virus RNA is a helix in the middle here with this nucleocapsid protein bound around it. And the nucleocapsid protein, unlike the spike protein, doesn't mutate so much, it's more conserved. However, it doesn't really induce this protective antibody so much. So I'll come back to those points. Can I have the next slide, please? Right, so in terms, I'm going to start with the tests first of all. And again, a lot of people listening will be familiar with ELISA tests. And these are really the kind of tests that are used in pregnancy tests, etc. And obviously in major industrial and research laboratories, that they're very important assays and highly commonplace. So the one I'm showing here is the capture ELISA, and the more basic one doesn't have the capture step. And this is where you coat the tray or the well with an antibody which recognizes your target antigen. So let's just say in this case it's the virus spike protein is your antigen. The capture antibody will recognize that and hold it in place, and it can actually purify it to some extent and hold it there. That's your target. And then if you're testing for antibody, somebody's blood, etc., to see if they've got antibody against the virus, this is then put on top. That antibody also will recognize the target antigen. And then we have to have some way of reporting that. So in an ELISA, it's an enzyme which is linked to what's called the secondary antibody. Usually there's another antibody on top, and then we can measure that with an enzyme substrate reaction and get a coloured product. So that's the basic of the ELISA test. You can also have a similar test where your reporter is of a different kind, maybe a fluorescent molecule, so you can actually see the fluorescent labeled product, and particularly you may have to excite that to see it with a UV or a laser. So that particular type of antigen antibody test is the basis of many of the tests that are being used. And in this case, you're trying to find antibody, but you can do it the other way around, where you use antibody and then you're looking for antigen. Will I have the next slide, please, Jonathan? Thank you. So the people will have heard a lot about lateral flow tests by now. And these are basically like ELISA tests, only they're done on a piece of nitrocellulose, and the reagents actually flow up the nitrocellulose where the reaction takes place. Now, early on, much earlier last year, everybody was talking about whether they had antibody to the virus, whether they were immune, and they were sold these lateral flow tests commercially. You could buy them in Boots, etc., and other companies, and people wanted to know whether they were positive. And in these tests, you would take a pinprick of blood and put it in some buffer that would come with the test. It would then go in the well there, marked S, and that would flow on up the nitrocellulose, and then it would meet antigens which have been put on and have been put there to actually capture the antibody coming in, and it can be arranged that you can pick up different kinds of antibody depending on where the test has been set up. So the particular test measured what's called immunoglobulin G or immunoglobulin M, and the control would pick up all antibody. So you can we're going to see a positive result, and if you had a positive test. So that was the lateral flow for antibody, and that's from one company, Abexa, that we're selling them, and that these can still be used. The problem is that they're not that sensitive. You need reasonably high levels of antibody, and also obviously if the user doesn't do the test correctly, then there can be problems. And if you get faint lines, what does that mean? Are you positive or negative? So the interpretation might be difficult. Can I have the next slide, please, Jonathan? Right, so the actual gold standard test that was being used in labs and the government approved is much more sophisticated, and this was products, Roche products, and also their big Cobas e-analyzer was required to do this test. Why is this much more sensitive? It used what's called electrochemoluminescence, and how this works is that their test actually didn't use the spike protein, it used the nucleocapsid protein. So they were looking for antibody to nucleocapsid protein. And obviously this was fine when we were looking for whether people had actually had the virus infection or not, because they would have been producing the virus nucleocapsid protein as well as the spike and other proteins. So in this case, they used the element eubinium, which is an alkaline metal, and therefore conducts electricity, and that was linked to the nucleocapsid protein. this was then used in the machine, and the antibody at the patient's sera were tried on this to see if there was actual capture, and if you actually got capture on the antigen was captured by the antibody, then you would get a reaction taking place, and the electric current would then produce this luminescence. And so this was a highly sensitive test. At the time when they were setting this up, they used 40 potentially cross-reactive samples to see not only was it a sensitive assay, but it was also highly specific. So they used from a whole range of coronaviruses, human coronaviruses and other animal coronaviruses, which had been confirmed initially that the samples were correct by PCR, and they actually showed that this test was highly specific. So this was the official government really testing that was done, and the labs that were all doing antibody testing would have had to have this particular machinery. Okay, can I have the next slide, please? Now, the other thing that is crucial, either in the infection or obviously after vaccination now, is testing at the right time. I mentioned earlier that the different classes of antibody and the two types, there are other types, but the two types relevant here are immunoglobulin M, which is only produced very soon after infection in the first 1 or 2 weeks, and then it drops down again. And the second type is immunoglobulin G, which takes longer to come up, at least 2 to 3 weeks. And then it doesn't go away completely, it'll drop a bit. And then when you either see another wild type infection of the virus, or you become vaccinated for the first or second time, the immunoglobulin G will boost further. And this is the basis of the vaccine, why you're getting a booster dose as well. But you can see if you actually test somebody for antibodies in the first week or so of infection, you're not going to find them. So you could get a false negative. And this is why these lateral flow tests, people, when they're taking them and when they're actually doing the test is so important, that if it's too early, and again, if antibodies drop down very low, then you're again not going to pick them up, particularly by a lateral flow test. So the sensitivity is crucial, and the timing. Can I have the next slide, please? Thank you. So the other test, this is still a lateral flow test, but this time this is the antigen test. And this is the one there's a lot of talk of at the minute that they're using in schools in England and in nurseries and other places, and also at airports and things like that. This is using the same principle of the antigen antibody binding again, but it's done the other way round. Now in this case, you're doing a nasal swab, that's going into buffer, and then that is going onto the lateral flow test. That flows on up, and it meets antibodies to the virus that have been put on the membrane. And then if you have a positive result, in this case, you'll get a color reaction, and this is done in various ways, either with an enzyme or with colored gold-colored beads, so you can actually see the reaction. These tests can also be done with saliva, and I believe that's reasonably accurate and works quite well. But the official government ones are done still with a nasal swab, not the throat, but just the nasal swab. So these tests are again, when you take the test, obviously, if somebody is hasn't got enough antigen there, i.e., if they actually have been infected, but very recently, so there's not enough virus there, these tests won't pick it up. It's not sensitive enough. Or if you're still producing some virus, but at very low levels at the other end of the infection, again, they may not pick it up. So they're only about 60 to 70% maybe accurate, but they're better than nothing if you're trying to pick up individuals, say in a school, etc., who may be positive. Right, can I have the next slide, please? Right, so I'm going to then move on to kind of the molecular tests looking for viral RNA. And many people are familiar with polymerase chain reaction. I'll just mention it briefly in case people aren't. But basically, this is where we're looking for small amounts of DNA, and it can be used for RNA, and I'll explain in a minute. So we're from DNA, which is double-stranded, that it has to be denatured to split the hydrogen bonds in the DNA, and that's done by heating up the DNA. And then if we're looking for a particular sequence, like the virus spike protein or part of that sequence, you have to have what are called primers, which are designed against the virus DNA sequence, so as we actually need to know a little bit about the sequence to start with. So those two primers are then what's called annealed to those single strands of DNA, and then it requires a polymerase enzyme which then copies the DNA in between those two primers, so you get a new strand made. And so that new strand is produced, and the whole process then can be done again and again and again to give you amplification. So starting with one piece of DNA, you can produce hundreds, which then can become visible if you run them on a gel or something or other ways of visualizing them. Now you're saying, well, the coronavirus, as you maybe know by now, is an RNA virus, so it has an RNA genome, so it's not DNA. So for all types of RNA, if you're going to do PCR, you have to make a copy of the RNA into DNA using an enzyme called reverse transcriptase, first of all. So you basically take the RNA, you copy it into a DNA form, and then you can do your PCR. Can I have the next slide, please, Jonathan? Thank you. So what's being done for then in the COVID tests is more complicated, and this is real-time or quantitative PCR. This is routinely used in diagnostic labs, now medical and veterinary, etc. So this is just slightly more complicated. You still have your two primers. You now have what's called a probe, which is a slightly longer piece of DNA, which recognizes a sequence in between the primers. The probe is labeled at one end with a fluorescent molecule, and at the other end a molecule called a quencher, which actually doesn't allow the fluorescence to take place. It stops it. And what happens there then, again, you're starting with your viral RNA, you do your reverse transcription to get DNA, you put your primers on, and the probe, and they will then, if the primers hit the viral sequence, and the primers will, the polymerase enzyme will then copy along, and the enzyme has another function. It has what's called an exonuclease on it, and once it hits the probe, that exonuclease reaction will actually snip off the quencher, and the fluorescence will then be visible. So these, again, this you can do lots of cycles, and the number of cycles that it takes place to actually see the fluorescence will depend on how much viral RNA you have there to start with. So if you have a lot of viral RNA, you'll need less cycles. If you have very small amount, you'll need more cycles. So the lower the cycle number, the more viral RNA you have. So that's the basis of the quantitative PCR, and obviously it can be read mechanically. You don't need to visualize that by eye. That's all captured, and TACMan is one of the major companies that do this. Next slide, please. So I was talking about the parallels with human and veterinary medicine, and just coming on to our own, why we have come into the equation, working with the Queen's AFBI and UU Alliance. At AFBI, we do veterinary diagnostics, and in virology, we have a molecular diagnostic lab, which routinely does qPCR, and obviously in a very regulated and quality assured way, the same as the hospital labs. So we were asked to come in pretty early on to help the Royal to do a lot of testing. So we've been testing from very near the start of the pandemic, you know, a month or two in. It took quite a lot of work to obviously set this all up, but we're used to testing for all these diseases there. I'll not go through them well. You'll all recognize foot and mouth, which we don't have any around at the minute, but we do a lot on avian high pathogenicity strains of avian influenza, etc. And you may not be aware that we've just actually had a big avian influenza outbreak starting in January, which took up a lot of our time. So as well as COVID testing, we were having to deal with the avian influenza outbreak as well. We are accredited to what's called ISO 17025 accreditation, which is for veterinary diagnostic labs, but for medical diagnostic, it's ISO 15189, but they're very practically identical. So we were in a very prime position to take on the testing. And we've also had Queen's people up and trained those as well. So that was just converting our labs to do the COVID testing. Can you give me the next slide, please? Thank you. So I'm not going to go through this in detail, but there's another test which the quantitative PCR requires high throughput robotics, etc. Thermocycler machines, expensive thermocycler machines, because the temperature has to go up and down a lot for the different stages. A more simple assay, which has been used near the bedside in some hospitals, is called the LAMP assay, or loop-mediated isothermal amplification test. It uses much simpler machine. It's done at more or less a constant temperature of heating up. It uses a lot more of these primer molecules, and there's a lot of looping round of the DNA. And you have what's called loop primers. Rather than do by fluorescence, you can do it by fluorescence, but you can do it by a colored product, or even because you actually get you can make lots more copies of the DNA using this method. You can actually visualize that directly. It becomes hybrid, but you can also use molecules, something like cyber green, so you get a colored product. And again, how much of the virus is there at present, the more of the color that you get, the darker it becomes. So that has been used in the hospital settings, and it's a reasonably accurate test, but it's still not taken as the gold standard of the real-time PCR. Can I have the next slide, please? Right, so I'm going to then, that was kind of diagnostic tests. I'm going to move on to the vaccine side. So you see from this, there are, these are showing you all the different vaccine types out there, and I'll go through some of these in a minute. Some of them you'll already be familiar with, and you can see, I'm not going to go through it, but there are, I mean, several hundred either developed or being developed around the world globally. Obviously, they're not all through the regulators. Only a few have come through at the minute. But the coming from the simplest types, the inactivated vaccines are a general kind of more old-fashioned type that we'd be aware of, where you just take the organism and you kill it, and then you shove it into somebody's arm. And that's being used effectively. We're using in that activated polio vaccine still in developed countries, you know, there are quite a lot of vaccines are inactivated in that way, and they can be very effective and usually have to be given with adjuvants. The next type, again, is an older method, the live attenuated, and a vaccine, in that case, the MMR vaccine, all the components of that, the measles, mumps, and rubella, would be live attenuated viruses. And they're usually attenuated by passing them through cell lines, etc. And it's a bit like Russian roulette. You select variants then that are attenuated and don't give you the full disease, but only a mild, or at least provide immunity. But they replicate like the normal wild type viruses. They're just attenuated. Subunit vaccines, again, we've had those around. An example of that would be the either the hepatitis B vaccine or the papilloma vaccine given to girls, and that's just using one kind of viral protein, a subunit. The ones which are newer are the viral vectors, which I'm going to go into. Now, they have been used quite a bit in veterinary medicine, but we're now using them for COVID in human medicine. DNA vaccines, which again are plasmids, which taking just DNA directly into the cell with the viral protein coded on it, and then messenger RNA, again, which are one of the types used for the COVID vaccine. So the viral vector one example is the Oxford AstraZeneca, and the messenger RNA, an example, is the Pfizer BioNTech, which are the three main ones being used in the UK. Just want to mention all these vaccines being made, how are they compared and regulated? Well, you may have heard of the Coalition for Epidemic Preparedness Innovation, or CEPI for short, and CEPI was set up in 2017 following the big Ebola outbreak, because realized getting vaccines through quickly, getting them made, produced, regulated was a major problem. So CEPI have taken this kind of under their umbrella, and they've now set up a centralized trial network of labs, mainly accredited diagnostic labs, which will, if people are making COVID vaccines, they will test the samples from either human trials or animal trials to make sure that everything is standardized and they can do direct comparisons. Next slide, please. Right, so I'm just going to explain a bit more about the two main vaccines being used in the UK and Ireland and wider in Europe. So the Pfizer BioNTech vaccine, which is very similar to the Moderna one that's being used in the US. I was in a call meeting with colleagues in the US this afternoon, and they had all been vaccinated with the Moderna vaccine. So basically, this is what's called a messenger RNA vaccine, and it's inside a nanoparticle, a lipid nanoparticle. So the messenger RNA is the molecule that we have them in our own cells, and these are the messengers which are made actually from our DNA and our cell nucleus. They copy the information from the genes there. They go out into the cytoplasm, and then they tell the ribosomes in the cytoplasm what proteins to make. So in this case, the virus spike protein messenger RNA is being delivered directly into cells. The lipid nanoparticle fuses with the cell membrane and releases the messenger RNA into the cell. That goes to the ribosomes in the cell where the spike protein is made, and it's done in it's a trimer molecule, the complete spike. So that is actually assembled in the cell, goes onto the cell surface, and then your immune system recognizes it. So that's the basis of the messenger RNA vaccines. Next slide, please. The AstraZeneca vaccine is using what's called a virus vector approach, and they're actually making what's called infectious particle vaccine. And I'll explain this a bit more now, but basically that you take a safe virus, one that you know doesn't harm humans, doesn't cause disease in humans, and into the genome of that you put the gene of your pathogenic virus, in this case the SARS-2 spike protein, into that. So you make a chimeric virus, which can then deliver the sequence to make the spike protein to the cells. Now, to make it extra safe, you can disable your vector, so it actually can't go on replicating. It'll get into the cells the first round, but it's disabled because it's missing one of its own proteins. Genes, and so it can't go on to continue replicating in further cells, but it takes them into the first lot. So this diagram's just showing you the snipping out of the sequence for the SARS spike protein. That's then put into the backbone virus, and in this case it's an chimpanzee adenovirus. These infectious we call them infectious particles because they're not fully replicating viruses, are then injected into the person, and those chimeric virus particles go into your cell, but they'll make the virus spike protein, but they can't go any further. Next slide, please. So those are parallels again with veterinary medicine here. Now, a lot of these vectored vaccines have already been used in veterinary medicine. Veterinary medicine is much more open to taking on board these new technologies, and probably because people perceive there's less risk in giving it to an animal than a human. But a lot of these are being developed, and in fact, the Oxford group already had made some veterinary vaccines using their process, and that's why they were ready to roll with the COVID one. So just parallel to our own work a little bit, with Professor Alton Power at Queen's, we're making some replicating vectored vaccines, but we're making both live replicating ones which will continue to replicate in the animal, and also some of these non-replicating particles, the same as the AstraZeneca vaccine. And it's just a diagram showing you that you're putting the gene in from your virus of interest that you want a vaccine against inside your particle. So we're making vaccine, well, we've actually made them, we're ready to go into trials with them, to bovine respiratory syncytial virus, which is actually very similar to human RSV, which is a major problem in babies. We're making one to 3, and we hope to make one against bovine coronavirus. We're also making one to a virus which is a problem in Southeast Asia, Nipah virus, but it's on the WHO list, and we got funded for that by Innovate UK to actually make that that vaccine. And Nipah virus infects pigs and humans, and again, it's another virus that came out of bats. Lots of these viruses come out of bats. So we actually have both the RSV and the Nipah virus vaccines ready now to go into trials. Can I have the next slide, please, Jonathan? Right, so just to point out, this is a slide I actually stole from Alton. The RC means the replicating vaccine. We're using a vector, a different vector to the Oxford group. We're using one called Sendai virus, which is a mouse para-influenza virus. It doesn't cause disease in humans or other animals. It's a mouse virus, so we can use it for our cattle and pig vaccines, and possibly for the human Nipah virus vaccine too. And as you can see in the first diagram there, we make the chimeric virus with the protein from bovine enterovirus or Nipah virus or parainfluenza, and it's inside the Sendai virus vector virus, and the vectored virus will continue to replicate like a normal virus if we give it to animals. And that might be good. It probably gives a much better immune response that way, and it might even pass to other animals, and we can give these vaccines by the respiratory route rather than injecting into your arm as well. So they're given by a natural route of infection, which is better for producing some other antibody in your mucosa, your respiratory tract. They're not the replicating what's called incompetent ones, or like the AstraZeneca. We've knocked out a gene in the Sendai virus, and that protein wouldn't be made. We actually give the protein separately in, so we get these particles. We get the full virus particle made, which will go into a cell. But because that gene is missing, the virus won't make that protein that's missing again, and so it can't go on to further cells. And okay, you can see a little diagram at the bottom. You sacrifice one for the other. The safety might go up with the replicating incompetent vaccine, but you're probably losing the amount of protein that can be produced, and probably going to lose the level of the immune response. So these vaccines are just going into trials, so we'll be able to look at the actual immune response. Next slide, please, Jonathan. Thank you. So just to point out who's involved in the work that we're doing on these recombinant, I say the Alton is we're using the Sendai virus for the respiratory syncytial, the para-influenza 3, and the Nipah virus at the bottom. We're also using another vector system, which is a dog virus called para-influenza 5, and this isn't a vector we have here. It's one from the University of Georgia, and we're working with Prof. Biao He there, he's actually put the same bovine respiratory syncytial virus protein in his vector, his parainfluenza 5, and we're going to be trialing both all these vaccines and actually mixing and matching the vector systems to see if we get a better response if we use a Sendai virus and maybe a parainfluenza to boost and so forth, because you do start to make antibodies to your vector vaccine, and that might be a problem. So you might have to change vectors at some point, and that might be something that hasn't been raised for the the COVID AstraZeneca yet, but it is a possibility that that might happen. You start to make an immune response against your vector, then that can be probably won't get a good reaction. Sorry, next slide. Right, so just the last part, kind of I was going to talk about mutations in the SARS-CoV-2 virus, because everybody, obviously we've seen these variants come up, and this is, it's not a kind of a one-hit, a variant suddenly arises. The virus is mutating all the time, and it's known that SARS-CoV-2 accumulates 1 to 2 single nucleotide mutations in its genome per month. But if you think of that, that's about half the rate of influenza virus and about half of the rate of HIV, both of which mutate more rapidly. And the reason, actually, although people are talking about all these mutants, it's not as bad as some of these other viruses mutating, is that coronaviruses have a correction mechanism. It's called a novel exoribonucleotase, and it will actually snip out some mistakes and some of the errors as it's replicating, which a lot of these other RNA viruses don't have. So that kind of slows things down again. But you can see the spike protein in COVID-V2, it has 1,275 amino acids long, and obviously each amino acid is 3 nucleotides, and so that's a quite a large amount that can actually mutate to change the spike protein. Sorry, there's some noise in the background. I'm not sure. Below, it's not, you're not meant to be able to see it, but that's kind of mapping all the mutations that have taken place in the spike protein so far. So you can see that, it's not just single events. There are multiple events happening, and not all the mutations will survive, because the virus can only mutate within inside a certain window. If it mutates outside that, it can't function. It will go beyond its own function. So it has a limited kind of restricted window, which it can actually mutate. Next slide, please. Right, so this was just to show you that flu is much more a problem in terms of mutations. It mutates those single mutations as well, but because flu has an 8-segment genome, its genome is in 8 different bits, unlike coronaviruses, which have a single each genome. With flu, if you can have 2 different viruses, showing the H3 and H2 forms going, one from a bird and one from a human, and both infecting a pig at the same time, you get what's called segment mixing and reassortment. And that's why we get these big jumps with flu. And a lot of these flu pandemics have come out of Southeast Asia, and particularly China, because of the close proximity of their farming, where they have birds, poultry, and pigs, etc., and humans all mixing together. So you can get those large jumps. So that's actually much more worrying in a way in COVID. It's just that we have got vaccines to influenza, and we can make new vaccines every year based on what we think flu viruses are coming along. And hopefully something like that can be done for COVID as well, that we'll be able to make new vaccines as we think fit. Next slide, please. So in terms of the mutations at the minute, these are some of the variants that we obviously know about. There's a UK variant which arose in Kent, South African, Brazil and Japan, there's California and New York. So these are ones which have, although mutations occurring all the time, these variants have some advantage and have taken off in the population. So we're actually seeing them, and they've come up, and the ones that we need to be aware of. So they're kind of the areas that we're at, that area in red there is that area DG14G. That mutation is found to enhance virus replication and virus load. So that's helping the virus to actually replicate more and produce more virus. The mutations if you see the receptor binding domain in the blue box there, the RBD, that's the bit of the spike protein that actually binds to the AC2 ACE receptor. And if that mutates, it can make the binding worse, but obviously the variants that will get selected are the ones that make the binding stronger. So those mutations will help the virus get into the cell more easily. And also if that mutates more readily, then we will have a problem with the antibody maybe blocking it because it'll have changed too much for the antibody to recognize it. At site 700 there, you'll see what's called a furin cleavage site. This is where the spike protein has to be cleaved by an enzyme called furin, which is found in our cells in our respiratory tract. Now, this is something which seems to have occurred to help the adaptation to humans, because in the bat sequence there is no furin cleavage site. So this mutation has occurred to help and is enhanced then getting into the spread in the human population. Next slide, please. This is just up for the purposes of saying, if we're looking at protective antibody, that's the slide I showed earlier, the ELISA measures total antibody, and it'll not tell you whether your antibody is protective or not. That's all the reason for actually showing that again. Next slide, please. The kind of antibody that's being looked at to see if we're protected against the virus or against the vaccines is called neutralizing antibodies, and these act to stop the virus infecting the cell. The classical one people think about is where it binds to the virus and stops it getting into the cell, but neutralizing antibodies can actually act at different stages of the virus getting into the cell, and also they can go into the cell sometimes and block internal steps. So they're not always at the the classical bind and stop binding to the receptor. It can be more complicated than that. But again, obviously, if the virus mutates the spike protein, then these antibodies may not recognize it and may not stop the virus going into the cell. Next slide, please. They're not going to go through this in detail, but how you do these neutralization tests are you take a standard amount of virus, you make dilutions of your antibody from your person, from the individual's blood. You'll mix those dilutions with a standard amount each time of the virus, and you'll have a control which has no antibody, and then you look to see how much the virus infection is reduced. And this can be done by what's called a plaque assay, where the virus produces these little holes or plaques in the cell where an individual virus has gone in and replicated and made a hole. So we're looking for a reduction of 50%, and the antibody dilution that gives us that 50% reduction is the titer of your antibody. And then we can do it by another, I'm not going to go into a quantal method, which is actually a bit easier to do. Now, these tests, you either have to use the live SARS-2 virus, and that a lot of that work is being done in Porton Down, or you can use what's called a pseudotype virus, which is another virus, again, vectored virus, where you put the SARS spike protein in it, and you can use that to do your neutralization test rather than. So This is the kind of test that has to be done, and the CEPI central labs are doing this too. And this is a test that will be constantly being done to look at whether these variants that are arising are escaping our antibody response. So this is the important test to look at that, the neutralization test. Next slide, please. I think we're coming. So again, just to remind you, coming to the end here, that a lot of talk about antibodies and antibody protection, but it's not the only type of protection. The other side of the coin, for those of you who are immunologists, obviously know there's the cell-mediated response, killer cells and cytotoxic T cells, which, whereas antibody will neutralize virus in the blood, etc., externally, infected cells are killed by these types of killer cells. And again, that is a specific reaction. These are specific cell reactions. Certainly the cytotoxic T cells recognize, and it's whether, again, our immune system, if the virus can escape that cell-mediated immunity as well. So we'll be seeing the spike protein, particularly for most of the vaccines, and actually whether your T cells actually then recognize that. But if it mutates too much, then that might escape. But it is a second line of defense and may give us further protection, hopefully, as well as the antibodies. Last slide, please. Right, so just to conclude what I've said, there are a range of assays in place for virus antigens detection, antiviral antibodies, and also for viral RNA. These assays have different sensitivities and reliability, but they're used for different purposes and settings. So you couldn't do a quantitative real-time PCR test in a school, or you couldn't do a large electroluminescence antibody test either in a lot of settings. So the lateral flow tests have their purposes, etc. But you have to accept that they're not as accurate, and there may be pitfalls with them. There are obviously a wide range of vaccines that are produced on different platforms globally. And just to say, the Pfizer, BioNTech, and the Oxford AstraZeneca have been shown now to be highly protective, even in older age groups, against severe disease. And unfortunately, it's taken a little while for our European colleagues to maybe come on board with that, or certainly for the AstraZeneca. There are a lot of questions still remain. How long will the vaccine protection or protection from natural infection actually last? We don't know. Coronaviruses tend not to give us long-term protection, as say something like measles, which is lifelong. How often will vaccination have to be carried out? We don't know yet. Maybe it'll be once a year thing like flu. How many people have to be vaccinated to give herd immunity? That's not really known yet. It may be around 85%. And obviously there's some individuals in our community can't have the vaccine and others who won't have the vaccine. So the herd immunity will help to protect them. Again, do vaccinated individuals shed virus? I think that's being looked at very specifically now. But that's why we're still, even with being vaccinated, we have to go around wearing face masks still, because we don't actually know. Will variants arrive which the vaccine doesn't protect against? Again, we don't know. We know there is less protection from these neutralization tests to things like the South African variant, etc. So we know it is reduced, but it's still giving some protection. What's reassuring is because the platforms that are being used, the Oxford, the chimpanzee adenovirus, and also the messenger RNA platforms, which have all been through regulators, they rapidly can slot in these new variant sequences of the spike proteins that arise, and they'll become through even much quicker than the current vaccines. So that's going to be very, very helpful in getting these vaccines out there quickly. And in fact, Oxford, I know they've already started to make some vaccines against the new variants. So I'll leave it there. I hope I haven't gone on for too long, and happy to answer any questions at all on the subject.